Pseudogenes are considered by Neo-Darwinists to be one of the strongest evidence for Neo-Darwinian evolution, since those genes presumably come from a common ancestor and are presumably vestigial, meaning that they show little to no function at all and serve no useful purpose, unlike protein coding regions. Such arguments, however, are circular and really based on ignorance. So let's get started. There are three types of pseudogenes in the genome. One type is known as disabled pseudogenes, which are basically single sequences that may have once coded for protein but have apparently been inactivated by nucleotide changes or deletions known as mutations. A second one is known as duplicated pseudogenes, which are basically copies of still functioning genes, though unlike the functioning originals, they have characteristics that prevent them from encoding proteins. These type of genes usually arise from the process of unsuccessful gene duplication. The third one is known as processed pseudogenes, which basically have sequences similar to those of functioning genes, except that they lack promoter sequences as well as introns. However, the majority of pseudogenes fall in the category of processed pseudogenes. So in this video, we're going to rely on processed pseudogenes as opposed to the others. Now, neo-evolutionists claim that pseudogenes are very strong evidence for evolution, since they are vestigial and serve no useful purpose for the living organism. However, this is actually false. Starting in the early 1990s, molecular biologists have discovered that pseudogenes actually play an important role in what is known as RNA interference, where these RNA interference are basically RNA molecules that play a regulatory role in gene expression from the corresponding gene. In addition, molecular biologists have also discovered that the antisense strands located in some pseudogenes have the ability to transcript DNA nucleotide-based sequences into RNA, resulting into double-stranded RNA. To demonstrate the point, let's look at an actual illustration. If you notice here, there is a pseudogene as well as a parental gene. Both of these actually have something in common, and that is that they both share very similar DNA nucleotide-based sequences of G, T, A, and C. Now, if you notice here, the sense strand of the parental gene actually converts the nucleotide-based sequence of the GGTACAATTG into RNA. At the same time, you will also notice that the antisense strand located in the pseudogene actually converts its nucleotide-based sequences into RNA as well. In addition, the nucleotide-based sequences that is found in the double-stranded of DNA as well as RNA are actually complementary to each other. For example, A always attaches to T, while G always attaches to C. In RNA, however, there is an exception. Instead of there being thiamine in the RNA molecule, it actually gets replaced by uracil. So in this case, adenine actually attaches to uracil, not thiamine. The only way thiamine gets attached is if it was located in a double-stranded DNA. However, since this is an RNA strand, it would attach to uracil rather than thiamine. Now, if you also notice, RNA also has the same complementary model as the DNA double-stranded DNA. The only difference, however, is that RNA only uses uracil rather than thiamine. Since the RNA nucleotide-based sequences CCAUCUUAAC and GGUACAAUUG have binding together, they are considered double-stranded RNA. Since pseudogenes as well as their corresponding genes are capable of containing double-stranded RNA, their expression does not translate into proteins. However, what it does instead is it interferes with the translation and thereby reduces or regulates gene expression. Cells make good use of RNA interference to regulate the amount of protein they produce. So, it has found that pseudogenes are actually quite functional in the sense of RNA interference. Some pseudogenes contain double-stranded RNA, which result into the regulation of other genes. So much for the evolution fairy tale claim that pseudogenes serve no useful purpose in the genome when they actually do. Not only are pseudogenes found to regulate gene expression, but they are also found to play an important role in embryo development as well as the effectiveness of a protein. Now, another argument that Neo-Darwinists use in defense of junk DNA of pseudogenes is that if you knock out a sequence of a pseudogene and it doesn't have any function at all, then you can conclude that it is truly non-functional. However, this argument fails since it does not address all the aspects of possible functions. It only addresses a certain aspect of a function. For example, suppose I knock out a pseudogene and I expect there to be an effect in the embryo development or phenotype if it truly serves any purpose at all, and yet I find nothing. Does that mean that the pseudogene is truly non-functional and junk? No. What it means is that the pseudogene, whatever importance it has, is actually unrelated to the embryo or phenotypic stage. It is quite possible that the pseudogene, for example, can perhaps serve a useful purpose in adaptation in response to an environmental stress environment, where this adaptation response is activated in advance and beforehand, as well as predetermined and induced. Ironically, there are actually scientific studies that demonstrate so-called pseudogenes known as cryptic genes that actually play an important role in adaptation. Cryptic genes, DNA segments that give the appearance of pseudogenes but do not code for proteins and remain inactive, have been found in most bacteria and possibly in higher organisms. At least 90% of the bacteria of E. coli have cryptic genes for B-glucoside sugars, which was established by Hall and Betts in 1997 as well as cited by Hall in 1999. These cryptic pseudogenes are also found to play an important role in the synthesis of enzymes as well as amino acids. Cryptic genes are also known that code for antibiotic resistance in bacteria. This was published by Hall in 2004. These cryptic genes are generally found on plasmids, and when no antibiotics are present, the cryptic genes are silent, meaning that the resistance is not expressed in the microorganism. In the presence of antibiotics, an epigenetic change has occurred, turning off the signature and permitting the cryptic resistance genes to be expressed. This means that the antibiotics we administer against bacterial infection are reducing their own resistance, rather than mutating them through random mutations in the process of Neo-Darwinian mechanisms. Note that these epigenetic changes are regulated, occurring when the organism is being stressed by its environment, which was published by Hall in 1999. Hall described the importance of insertion elements in adaptively modifying the genome. Quoted, "Not only do IS elements make a direct contribution to fitness by activating cryptic operons, they do so in a regulated manner, transposing at a higher rate in starving cells than in growing cells. In at least one case, IS elements activate an operon during starvation only if the substrate for that operon is present in the environment. The activation of a cryptic gene is an example of a pseudogene as well as a genetic rearrangement responding to environmental inputs to make the organism more adaptive to the new environment." So basically what these studies show is that pseudogenes do not only play a role in gene regulation, but they also seem to play a role in adaptation in response to environmental stress, where these responses are activated in advance and induced non-randomly without even invoking mutations in the first place. Interesting how when species are exposed to an environmental stress stimuli, their genomes actually turn on genes non-randomly that was once thought to be non-functional and useless, pseudo, when they actually do play a significant role in events for induced adaptation. Wait, I thought there were pseudogenes that served no useful purpose according to the theory of Neo-Darwinian evolution, and yet it turns out that pseudogenes are actually quite functional in gene regulation, gene expression, as well as epigenetic adaptation in response to the environmental stress stimuli. How embarrassing. Now let's move on to the famous argument, vitamin C. Neo-Darwinists use the vitamin C argument as evidence for evolution and common ancestry. Ironically, if two or more species are not thought to be closely related, in other words, evolutionary cousins, through common descent, the pseudogene similarity implies independent parallel preservation by natural selection and not by inheritance of a common ancestor. And yet, if they are closely related through common descent, then the same pseudogene similarity is implied to be very strong evidence for common descent. Funny how evolutionists use similarity to conclude non-common descent when it comes to unrelated species, but use the same similarity but in different logic in favor of common descent when it comes to what they think are closely related species. Same evidence, two different contradictory conclusions. It's pretty clear that the so-called evidence of evolution by common ancestry is evoked only if it makes sense. Too bad it begs the question between independent conservation of natural selection and inheritance by common ancestry. So, to sum everything up, pseudogenes are actually not as useless as Neo-Darwinists have thought. They have significant roles in RNA interference, gene regulation, expression, as well as the advanced adaptation of species in response to environmental stress stimuli through epigenetic changes. Now, another interesting to point out is that the evidence for common descent with the arguments that Neo-Darwinists use when it comes to similarities to pseudogenes is actually based more on circular talk, which begs the question between conserved DNA, between natural selection, as well as inheritance of a common ancestor. Thanks for watching.